Bismillahirrahmanirrahim. This is the part 2 of chapter number 2, Cytoplasm and its organelles. Cytoplasm of the cell contains the basic and functional units of the cells and fat droplets, glycogen granules, vesicles, etc. But we have the five most important cellular organelles which we will discuss one by one. They are endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, peroxisomes. We will discuss one by one. Now, the endoplasmic reticulum. Endoplasmic reticulum. The endoplasmic reticulum, it is a membranous network, which is dispersed throughout the cell, This organelle is more extensive in those cells which are highly secretory in nature. Now, this is the structure of the endoplasmic reticulum is like this. The tubules, this is one tubule, this is another tubule, this is another tubule, this is another tubule, and like this they form an extensive network of tubules throughout the cells. There are two types of endoplasmic reticulum. One which has ribosomes on its surface. This endoplasmic reticulum is called the rough endoplasmic reticulum, and another endoplasmic reticulum which is smooth, it is called the smooth endoplasmic reticulum. The rough endoplasmic reticulum is responsible for the production of protein because of the ribosomes, which are machineries for the proteins. In the smooth endoplasmic reticulum, which contain different enzymes which are responsible for the production of fats. So, the endoplasmic reticulum is actually a highway, like the roads. This is the highway of the cell. Everything which has to go to the cell, it will must pass. First through this endoplasmic reticulum and then to the required area. And if everything which has to pass outside the cell, it will must pass first through this structure and then they will go outside the cell. So the endoplasmic reticulum acting like a highway of the cells for the incoming and outcoming things or compounds of the cells. Now this was the endoplasmic reticulum. Now the Golgi apparatus. The Golgi apparatus is stalk layers which is present to one side of the cell. These are the stalk layers like this. Network of stalk layers which are stalked over each other, and there are 4 to 5 layers which are stalked, and it also contains vesicles. The vesicles which are budding off from the Golgi apparatus. Golgi apparatus. So this is the Golgi apparatus. If anything which is synthesized endoplasmic reticulum will pass to the Golgi apparatus, and the Golgi apparatus will modify those things through the hydroxylation or phosphorylation, etc., and they will pack those substances which are released by the endoplasmic reticulum. So the Golgi apparatus acting as a modifier and packaging of the substances which are budding off from the endoplasmic reticulum. So and they modify these substances, and then they send these substances through these vesicle to the area to those areas which need these substances, whether it is outside the cell or inside the cell. So this is simply the Golgi apparatus and its structure, and this structure is similar in structure to the physically they are mostly similar to the structure to each other, but the Golgi apparatus is present to the one side of the cell. Now this was the endoplasmic reticulum, Golgi apparatus. Now we will discuss the lysosomes. The lysosomes, the structure like these are dispersed in the cytoplasm. The lysosomes. The lysosomes are these are actually the digestive structures of the cells which contain hydrolytic enzymes. Each lysosome is about 250 to 370 250 to 750 nanometer in diameter. These lysosomes further contain small granules, and these granules which are present inside this lysosome are 5 to 8 nanometer in diameter, and these granules inside contain specific hydrolytic enzymes, and there are about 40 different types of hydrolytic enzymes which are present in these small granules. What is the mechanism of hydrolytic enzyme, in which mechanism and how they are acting? The hydrolytic enzyme, for example, they through this is the water. Let's suppose this is the water, hydrogen. Their function is to break up this into hydroxide and hydrogen group, and then they add hydrogen to one side of the molecule and hydroxyl to the other side of. For example, if this is the compound, they will attach hydroxyl to one side and hydrogen to the other side, and will break this bond through the hydrolysis of water. So that's why they are called hydrolytic enzymes. These lysosomes contain hydrolytic enzymes. If in any condition these hydrolytic enzymes comes out of this lysosomal structure, then they will digest the cells. So lysosomal membrane is very highly stable to prevent the release of these hydrolytic enzymes. If this is the bacteria, virus, or carbohydrate, protein, if it comes through the phagocytosis, then it will be first engulfed by this lysosomal structure and will digest. And the digestive product will then be utilized by the cells, and the substances which are not digested remain as a residual body inside these lysosomal structures. So this was the lysosomes. Lysosomes are actually formed from these budding of vesicles of the Golgi apparatus. Some structures of this vesicles become secretory enzymes, some become the hormones, neurotransmitters, etc., and some become the lysosomes. So this was the lysosome. Now, peroxisomes. These are the peroxisomes. So if we take a look to the through the lysosome and peroxisome, they are similar in physical structure. But there are two conditions in which the peroxisome and lysosome, they are different from each other. First, the lysosomes are budding off from the Golgi apparatus. If we differentiate the peroxisomes and lysosomes. So how can we differentiate? First of all, the peroxisomes are budding off from smooth endoplasmic reticulum, or they are self-replicating organelle of the cell. And lysosomes, they are budding off from Golgi apparatus, Golgi apparatus. Second difference is the peroxisomes contain oxidases, contain oxidases, and the lysosome contain hydrolyses. Hydrolysis enzymes. The oxidases enzymes, these enzymes which I have explained what is the hydrolysis enzyme. The oxidases enzyme, these enzymes acting on the substances which are poisonous to the cells and oxidize those and form the hydrogen peroxide. They form the hydrogen peroxide by oxidizing different substances in the cells. So and in association with another enzyme which is called catalase, in association with um, oxidases, oxidases and plus catalases form hydrogen peroxide and prevent the cells from the damaging effect of any poisonous substance. So this is the difference between the peroxisome and lysosome, and physically these both structures are different, are similar, but in these two aspects they are different from each other. So this was the lysosome, this is the peroxisome. Now one important organelle remains: mitochondria. Mitochondria. Mitochondria is also called the powerhouse of the cells. First of all, we will draw the structure of mitochondria. Let's suppose this is the mitochondria. This is the outer membrane of the mitochondria, which is smooth. The inner membrane of the mitochondria it is present in the form of the shelf-like structures. The inner membrane of the mitochondria is present itself in a shelf-like structures. These shelf-like structure, these are called cresti. What are they called? Cresti. These cresti contain the oxidative enzymes which are necessary for the Krebs cycle. Sorry, for the electron transport. These cresti are loaded with oxidative enzymes which are necessary enzyme for the electron transport chain. So this is the outer membrane, outer membrane, inner membrane, inner membrane, and the space between the outer membrane and the inner membrane is called the intermembrane space. Intermembrane space. This is the intermembrane space, and this space, the space which is inside the inner membrane, this space contain the matrix, and the matrix contain different types of proteins, enzymes, and the mitochondria, you know, it also contain its own DNA. So the mitochondria do not form from any structure, but the mitochondria is a self-replicating organelle of the cell and contain its own DNA. This matrix contain the enzymes which are necessary for the most of the important enzymes which are present in the space. These are the enzymes which are responsible for the Krebs cycle. And the matrix also contain proteins and DNA. The enzymes which are necessary for the electron transport chain, they are present in these shelf-like cresti of the inner membrane. So this is the structure of the mitochondria. So what is the function of the mitochondria? The function will be discussed in the upcoming in the upcoming discussion, but we will take an overview. The glycolysis occurs outside the cell. Glycolysis, which occurs in the cytoplasm, they form the pyruvic acid. The pyruvic acid then comes to the to the inside of the matrix. Then the matrix through the Krebs cycle convert this pyruvic acid to the NAD and FAD, NAD and NFAD, NAD and FAD. Then these are catch up by these shelf-like structure, and through these oxidative enzymes, they convert the NAD and NFAD to energy currency, which is called ATP. So this is an overview. We will discuss um, the functional machinery of the mitochondria in the upcoming discussion. So these were the five most important organelles which I have discussed. First of all, the endoplasmic reticulum, then Golgi apparatus, then lysosomes, then peroxisomes, and then mitochondria. So this is done, guys. The organelles are done. So if we take a look, the endoplasmic reticulum is done, the Golgi apparatus, the lysosomes, the peroxisomes, the secretory vesicle. I didn't discuss the secretory vesicle, but the secretory vesicle are in high quantity in those cells which are secretory in nature, and these are simply which I have discussed in the endoplasmic reticulum, that the secretory vesicles are budding off first from the endoplasmic reticulum and then goes to the Golgi apparatus for packaging and modification. And these secretory vesicles may contain hormones, enzymes, or neurotransmitters depending on the type of cells. So the secretory vesicles are highly in a high community in those cells which are secretory in nature. So this was the secretory vesicles. Mitochondria. And mitochondria, I discussed it. One more important point, that the mitochondria are present in high quantity in those cells which are highly active. For example, the cells of cardiac muscles, the skeletal muscle cells, etc. These are highly active, so they contain a large number of mitochondria inside them. Now, cell cytoskeleton, filament and tubular structures. We will discuss the cell cytoskeleton. So what is actually the cytoskeleton of the cell? The cytoskeleton is actually the framework, like the framework which is for our body. The bones of our body act as a framework for the human body. Same is the case for the cell. These filamentous structures which are present in the cell act as a cytoskeleton for these cells, and they maintain the shape, the function, and integrity of the cells. So the cytoskeleton are very important in these aspects. First of all, we will discuss the microtubules. The microtubules it is actually found by the tubulin molecule, alpha tubulin and beta tubulin. These alpha tubulin and beta tubulin molecules, they form a circle, and this circle can contain up to 13 units of alternating alpha and beta units. So in this way, they are adding to each other. In this way, they form circle one by one, and in this way, they form a tube-like structure. And each circle contain one tubulin molecule, one alpha tubulin and one beta tubulin. And in this alternating way, they form a tube-like structure, which is 25 nanometer in diameter. So the microtubule has many important functions of the cell. Microtubule and its function. The microtubule is responsible for the structure, for the formation of cilia. It is responsible for the formation of flagellum. It is responsible for the formation of the structure of spindles of centrioles. The microtubules also maintain shape and integrity of the cells. Number 5, microtubule act as a belt which carry one molecule from one side of the cell to the other, which carry one substance from one side of the cell to the other. So this was the functions of the microtubule. There are a lot of many functions, but I have discussed few. Now the microtubule. Now we will discuss the intermediate filament. This is the intermediate filament. This is the intermediate filament. The intermediate filament is formed from the protein dimer. The protein dimer structure wrap around each other and form this dimer-like structure. This is simply smaller than the microtubule, and it is 8 to 12 nanometer in diameter. But average we having the diameter of the microtubule, the intermediate filament is 10 nanometer. 8 to 12 is the range, and 10 nanometer is an average diameter of the intermediate filament. Intermediate filament are actually the intermediate filament are easy in its function. The intermediate filament, it's only maintain the integrity, shape and flexibility of the microtubules and support the microtubules. This is the intermediate filament. Now the microfilament. The microfilament, one microfilament is made up of the G-actin molecule. The G-actin molecule, one F-actin, I will show you like this. This is, if for example, microfilament. The microfilament are made up. One microfilament is made up of two F-actin molecule which wrapped around. If this is one F-actin molecule and this is another F-actin filament. This is one F-actin filament, another actin. The two F-actin filament wrapped around itself and form the F-actin filament. And this one F-actin monomer is made up of what? One F-actin filament. For example, the G-actin filament. This is one G-actin filament, another G-actin filament, and this G-actin filament are attaching to each other. This is G-actin molecule. G-actin molecule. This is one G-actin molecule. This is the F-actin filament. The G-actin molecule combines together and form F-actin filament, and in this way, two actin filament. This is one actin filament, and this is another actin filament. They wrapped around itself, and in this manner, the two F-actin filament wrapped around itself and form the F-actin filaments. So the F-actin filaments are present below the cell membrane, and it acts as a supporting structure for the cell membrane, and it is present in the apical region of the cell membrane, which is called the exoplasm. So this was about the microfilament. Another thing which is important about the intermediate filament, that the intermediate filament, they are called differently according to the cell they are present. For example, in the neurons they are called neurofilaments, in the skin they are called keratin filaments, in the muscle they are called desmin filaments. So in according to the structure and according to the cell they are present, the intermediate filaments have different names. Why they are called intermediate filaments? Because the diameter of the intermediate filament is between the actin and myosin filament. Actin is a thick protein, actin is a thin protein, and the myosin is a thick protein. So in between them, their diameter is present. That's why they are called the intermediate filaments. Now the cytoskeleton is done. Now the nucleus. The nucleus is the most important structure of the cell. The nucleus controls all the metabolic functions of the cell through its genetic machinery. For example, if protein is required, if lipid is required, if hormone is required, if neurotransmitter is required, then for all these functions, the main controller center is the nucleus. If the nucleus is not functioning, then the cell will die and it cannot survive, because the nucleus, through its transcription of DNA, it sends the messenger RNA to the cytoplasm, and through this messenger RNA, different protein and structural protein, enzymatic proteins, and different enzymes which are required for the neurotransmitters, and enzymes which are required for the lipids, and different substances are synthesized to this messenger RNA. So the nucleus is actually the brain of the cell. If someone lost his brain, then he will eventually die. He cannot survive. So this is the brain of the cell. The nucleus is called the brain of the cell. The nucleus. The nucleus is surrounded by a double nuclear membrane. For example, if this is the nucleus, for example, This is the nucleus, and this is the cytoplasm. This nucleus is surrounded by nuclear envelope, which is a double layer membrane. For example, this is the double layer membrane. A double layer membrane. Why I have left these pores? Now I will explain why. These are the pores. These are the nuclear pores. These are the nuclear pores which receive and send different substances, and is necessary for the for the exchange of different substances between the nucleoplasm and cytoplasm. These pores of the nucleus are actually these are in contact with what structure? What structure, you know, guys? Now this is very important. This is connected with the endoplasmic reticulum directly. So you got it. This is the nuclear membrane, and these pores between the nuclear membrane, the pores between the nuclear membrane, these are the pores. These pores are directly connected with the endoplasmic reticulum, with the smooth endoplasmic reticulum of the cell. And these nuclear pores are surrounded by the proteins which guided these pores, by highly organized structural protein which guide these nuclear pores and control the entering of the substance which are entering the nucleus and which are coming out of the cell of the nucleus. So these are the nuclear pores. This was the nucleus. This was the nucleus. This is the cell membrane. This is the nucleus. This is the nuclear membrane. The nuclear membrane contains the pores which are in contact with the endoplasmic reticulum. These pores are responsible for the exchange of substances between the cytoplasm and nucleoplasm. So this was about the nucleus. Now the nuclear membrane is done nucleoli in the formation of ribosomes. Now this is the dark staining organ. The nucleolus is the dark staining organ which is present in the nucleoplasm. This nucleolus is responsible for the formation of ribosomes. So from where the ribosomes comes, the ribosomes are synthesized inside this nucleolus. They are synthesizing the ribosomes. The nucleolus contain the same RNA and proteins which are present in the ribosomes. From where this RNA comes? RNA comes from the by the transcription of DNA. When the DNA is transcribed, then some of the RNA comes to the cytoplasm and some are stored in the nucleolus for the formation of ribosomes. So this is simple. The nucleolus is made up of the RNA plus proteins. This structure is responsible for the formation of ribosomes, and this contains the RNA of the same type which is present in the ribosomes, and this RNA comes from the transcription of the DNA. So this is about the nucleolus. Now part 2 of chapter number 2 is done. Guys, subscribe, like and comment my channel. Thank you.